Hello guys, welcome to my YouTube channel. So in this video, we are going to learn about the electron transport chain. So what is the electron transport chain? This is the process by which our body generates ATP through a series of complexes and some of the proteins located inside the inner membrane of the mitochondrion. Now let's see. This process is going to generate energy via oxidative phosphorylation. And remember, in my other videos, we learned about the substrate level phosphorylation, where we generated energy from glycosylic acid cycle, that is the Krebs cycle, and also from glycolysis. We generated 2 molecules of ATP from glycolysis and 2 molecules of ATP from TCA, or the tricarboxylic acid cycle. But this one just generates small amount of ATP. Now we want to see how the body generates a lot of ATP that help us do various activities, running, walking, doing all the other physical activities. Now let's see. Now remember I told you that you are going to use the NADH that is generated from the TCA, FADH2, and also the NADH from the glucose, the glycolysis, the glucose metabolism pathway. So this process, or these are the precursors that you are going to put inside the electron transport chain so that you can generate energy. Now let's get started. Now this one here, let me name them first. This is called complex 1, or you can simply call it NADH dehydrogenase, or complex 1. This one is called coenzyme Q. Coenzyme Q. Sometimes you can see it written Q10 in some books. Then this is complex 2, or succinate dehydrogenase. You can also call it succinate dehydrogenase or complex 2, just to make life easy. Then this is another coenzyme Q. Coenzyme Q10. Yeah, don't be surprised when you see it coenzyme Q alone. It's okay. You can just find some books Q10 or some books just Q. Now this is complex 3. You can call it coenzyme Q cytochrome B oxyreductase. Yeah, or complex 3. Then this is cytochrome C. Cytochrome C. Then this is complex 4. Also, you can call it cytochrome oxidase or complex 4. Now, let's see. NADH is going to get into complex 1. So, I'm going to have NADH. I don't know that can just put it there. So, NADH is going to come and unload the electrons it carries, right? So, remember it contains protons. So, the protons are going to be split into electrons and it contains hydride. This is the hydride. It's going to be split into protons and electrons. So protons are going to be taken now. So we have NADH. NADH is going to be oxidized to NAD plus. Right. Now it's going to give the electrons. So who is going to take the electrons? The electrons are going to be taken by a molecule called flavin mononucleotide, or FN. I'm going to say flavin mononucleotide. It's going to take these electrons and be converted into flavin mononucleotide with electrons. So FMNH2. So that you can see this very well is FMNH2. Flavin mononucleotide with electrons. Then it doesn't want that high energy state. Remember, the presence of electrons gives them high energy state, so it doesn't want the high energy state. So what it's going to do? It's going to pass the electrons to coenzyme Q. So we have coenzyme Q. It's going to be converted into QH2. So this is going to pass over the electrons to Q and it will be converted back to flavin mononucleotide without electrons. Now, that is for NADH. When you get to complex 2, now we are not going to use NADH. We have FADH2, right? So FADH2 is going to unload the electrons. So we have FADH2. It's going to be oxidized to FAD plus, right? So it's going to unload the electrons. And who is going to take the electrons? The same molecule, flavin mononucleotide. So we have flavin mononucleotide. It's going to take the electrons, right? Then it will be converted to flavin mononucleotide H2, just like in this other case. Now, flavin mononucleotide in complex 2 is also going to pass the electrons to coenzyme Q, right? Because it doesn't want the high energy state. So you have coenzyme Q is going to take up the electrons. So flavin mononucleotide is going to unload the electrons Then it's going to take up the electrons so that we convert it to QH2. But there's something you must not forget, guys. I must show you this one. Remember, complex 1 has a certain acceptor order transport. Now this one, whenever you pass the electrons from complex 1 to enzyme Q, it's going to generate some potential energy and it's going to pump the protons that we remember here is a hydride. We only unloaded the electrons, so we have protons all over inside the cytoplasm. There are so many protons. And we are going to take advantage of this concentration gradient that we're going to create here, so as to generate energy. Now, the side concentration of protons inside the mitochondrial matrix. Remember, this is a mitochondria inside the mitochondrial matrix. So, whenever you pass the electrons from complex 1 to coenzyme Q, it's going to generate some potential and some energy that is going to pump the protons from inside to the intermembrane space. So, the protons are going to pass out. So, I'm going to have the protons all over here. Alright, so these are protons. And you can see complex 2 that it doesn't have the receptor, right? So we're not going to pump the protons. Now let's move from coenzyme Q. Now, this coenzyme Q is going to combine with the coenzyme Q. Well, sometimes you can just draw it to have one so that we can just pass the electrons from complex 1 and complex 2, then we just put them together in one coenzyme Q. So these are going to pass the electrons to that other coenzyme Q. Now, it doesn't want the high energy state. What's it going to do? It's going to pass the electrons to complex 3. Now, complex 3, also called cytochrome B, it has a cytochrome B, and cytochrome B is attached to ion 3 and ion 2, 3 plus, right? So, coenzyme Q is going to pass the electrons to ion 3. So, it's going to pass the electrons and be converted back to Q. Then ion 3 doesn't want the high energy state, so it's going to be converted to ion 2. Right. Now, let's move to cytochrome C. So, cytochrome C also has ion 3 ions and ion 2 ions. Complex 3 is going to pass the electrons. So, it's going to pass the electrons to ion 3, then be converted to ion 3 plus. That is, ion 2 is going to pass the electrons to ion 3. Then ion 3 converted to ion 2. But remember, I told you it has a receptor. Whenever it pumps the electrons to the next molecule, it's going to generate some of the energy that's going to pump the protons outside. Yeah, so the concentration here is estimated to be something like 1 times 10 to negative 9 millimolar. So there's a concentration inside the matrix of the mitochondria. So this energy is going to be used to pump protons from inside to the intermembrane space. So we are going to have protons outside there. Now, here's cytochrome C. Cytochrome C is going to pass the electrons to complex 4, right? Now, so complex 4 has cytochrome A plus A3. So it is cytochrome A plus A3. It also has ion 3 ions, ion 3 ions and ion 2 ions. Now, the ion 2 from cytochrome C is going to pass the electrons, then be converted to ion 3. Then ion 3 gets the electrons and is converted to ion 2. Now, we are stuck. Where are we going next? We don't have any other complex there. Now let's see what's going to happen. Who is the last electron acceptor? Atomic oxygen. Now we have oxygen there. Atomic oxygen is going to take the electrons, right? It's going to receive the electrons, and also we're going to take the protons, which are found inside the matrix. It's going to form water. So oxygen is the last electron acceptor in the electron transport chain. Remember, none of them want that electron, the high energy state. So electron or mean oxygen is going to take the electrons that combine with the protons to form water. Now, let us see what's going to happen here. Remember, when you pass the electrons, you're going to generate some energy which is going to use to pump the protons into the intermembrane space. So we have a lot of protons all over. Right. Now, so this is going to be, we're going to create a gradient, a concentration gradient. Remember, we have different concentration of protons inside the matrix and also in the intermembrane space. Sometimes we can say there is about 1 times 10 to negative 7 millimolar. So you can see there's a concentration gradient that has been created. So we're going to take advantage of that so that we can generate energy. Now, we have complex 5, mostly called ATP synthase. Mostly call it ATP synthase. Now, ATP synthase, now let's see what's going to happen there. We have protons here, then you have a receptor, right? So this receptor is going to facilitate pumping, it's going to pump the protons from inside the intermembrane space back into the matrix, right? So inside there's a molecule, a protein molecule called adenine 210. This adenine 210 is the one that carries the protons from the other end onto the other end. So it's going to take protons, push it over to this end so that it can get into the mitochondrial matrix, right? Now, whenever it does that, whenever it takes the protons, pass it over. So you see, it's going to do this and passing it over. So whenever it does that, it's going to rotate. Whenever it rotates, it's also going to rotate. So this can call this part of the ATP synthase, you can just call it rotor, because it rotates. Then you can say this is the rotor. So this rotation of the ATP synthase, right, is going to lead to generation of some potential energy. That potential energy is going to be used by the ATP synthase to combine or bring together the organic phosphate, right? Organic phosphate means bring together organic phosphate plus adenosine diphosphate or ADP. So whenever the protons are passed from intermembrane space by adenine 210 located in that receptor there, it's going to take and pass. So whenever it takes and passes like this way, it's going to generate it's going to rotate. Whenever it rotates, it's going to rotate also the ATP synthase, right? This one here. So whenever it rotates, it's going to lead to generation of some potential energy. That energy is going to be used to, just going to fuse them. We're going to fuse organic phosphate and ADP, and what we're going to have? We're going to have ATP. At last. Now, that's how we generate ATP. Now, all these processes occur inside the mitochondrial matrix and in the intermembrane phase. So, we go through this complex 1, coenzyme Q, complex 2, another coenzyme Q, which is going to take the electrons from this other coenzyme Q, then combine them, then going to pass them over to complex 3, cytochrome C, and then complex 4. Complex 4 is going to pass the electrons to atomic oxygen or oxygen, which is going to take the electrons, combine with the protons to form water. Now, we are going to take advantage of the proton gradient or the concentration gradient of the protons inside the matrix and also in the membrane space. Now, we are going to pass them over via that receptor there, and there's a molecule there or a protein called adenine 210. So this is going to pass them over like this. So whenever it passes them over, it's going to rotate. When it rotates, it's also going to rotate the ATP synthase. When ATP synthase rotates, right, it's going to generate some potential energy. That energy is going to use to fuse adenosine diphosphate and organic phosphate so that form ATP. Now, this process of ATP formation is by oxidative phosphorylation. It generates a lot of ATP that is needed to run various metabolic processes inside our body, or various other processes of physical, like we can run, we can now walk, we can do any other thing. Now, there's something I need to show you guys. There are something called uncouplers and inhibitors, right? So uncouplers, let's study uncouplers. So they don't couple the electron transport chain with the ATP synthase that can generate energy. So the first uncoupler is let me show this. Those people who are fat, they will want to desire to reduce in weight, right? They want to reduce weight. So some doctors used to give them some drugs. So this drug is going to create a pore. This drug is going to create a pore here, or a receptor. Just draw it this way. Right. So remember, this receptor here, let me give you its name first. It's called 2,4-dinitrophenol. Now, this receptor here is going to uncouple the electron transport chain with the ATP synthase, so that it's going to facilitate or allow the passage of protons from the intermembrane space inside the mitochondrial matrix. Remember, without generating ATP, because it's not connected to ATP synthase. So it's just going to facilitate protons moving inside. But remember, we need the protons to move by the ATP synthase so that we can generate energy in form of ATP. So it's not going to couple ATP synthase with the electron transport chain. So it's going to allow the protons to get inside to the generation of any ATP. Now when that does, the ATP levels in our body is going to go down. Now what's the effect? When it goes down, TCA and glycolysis, the process will run so fast because they feel like there's no energy in the body and we're producing the NADH, the FDH, so it seems like they are not enough. So they're going to run so fast, they're going to work so hard so that they can generate NADH and FADH so that they can be taken. But remember, the problem is here. They generate everything and ATP rate type of chain is working. But remember, you can't this tophenol uncouples the electron transport chain, the ATP synthase that can generate energy in form of ATP. So that has a very great effect because it's going to increase the temperature. When it increases the temperature, then these people will find a way or a mechanism of losing in weight. But it's not actually preferred because it uncouples and it can lead to other disorders. There are also other uncouplers like the FCCP. FCCP. Then there's also another one called trichlorophenol. Now this FCCP, I can just call it for just a very long name. You can say carbonyl cyanide, P, trifluoro, methoxy, phenyl, hydrazine, but this is so hectic. You can just call it FCCP, but that's the full name. So these are uncouplers. They don't connect, they don't couple the electron transport chain with the ATP synthase. So they are going to lead to high metabolic rate and going to generate temperature. Alright. Now there's also another one. Now remember, babies, the children which are maybe they are born or some few weeks after being born. Remember, they have something called a brown adipose tissue. Now what is that brown adipose tissue? The brown adipose tissue is the adipose tissue. It's only that it has a lot of mitochondria. Now it's the mitochondria that make it brown. Now, this brown adipose tissue secretes a molecule. This molecule is called thermogenin. Now, what is this thermogenin going to do? This thermogenin is going to create another pore here, just inside the intermembrane, the inner membrane. Right. Now, that receptor there is also going to do the same thing, just like the uncouplers, the 2,4-dimethylphenol, they are going to facilitate the penetration of protons inside the matrix without generation of energy because it doesn't couple the electron transport chain with the ATP synthase. So they facilitate the movement of protons from the intermembrane space inside the mitochondrial matrix without generating ATP. Now, this one of course has an advantage in terms of children because they need to remain warm. So remember, when this does, the ATP level is going to go down. When it goes down, we're going to have TCA and glycolysis running so much, and that this is going to high metabolic activity is going to happen. So this is going to generate a lot of energy inside the body, a lot of heat, and this is good because the children and babies need to remain warm, right? Now that also is important. Now something we need also to know, guys. We also have inhibitors. Now there are certain molecules or chemicals that inhibit the electron transport chain. Now, for example, at complex 4, remember we were passing the electrons from complex 4 to atomic oxygen to form water because it is the last electron acceptor. But there are certain molecules such as cyanide. Let me just write cyanide. So cyanide is going to cyanide. When someone has cyanide poisoning, to me, it's going to inhibit the passage of electrons or electrons moving from complex 4 to atomic oxygen. So that can lead to other complications. Another one is called carbon monoxide. Yeah, carbon monoxide. That one is also going to inhibit complex 4, passing the electrons to atomic oxygen. That can lead to some complications. Now, what else? There's also another one at complex 3. This is called antimycin A. anti-mycin A. Now, this one is going to prevent the passage of electrons from complex 3 to cytochrome C. So, that can also lead to other complications because none of these complexes here want that high energy state. So, that will force it to have the high energy state, and that can lead to some metabolic disorders. Another one is here, complex 1. So, there's also a molecule called amita. Another one is called rotinone. Now these are inhibitors. They inhibit the flow or the consistent flow of the electron transport chain. So they disrupt the flow of electrons, and therefore, and then we not have the ATP. And remember, we need ATP so much for various processes in our body, like the moving, burning, breaking down, and catabolic reactions. They need energy. So without ATP, that will be off. Now what else do we need to know, guys? Oh, there's one thing you need to know. Remember, we input NADH complex 1 oxidized to NAD+, right? So remember, we also generated some NADH from glycolysis. Now we didn't see how those ones get into the mitochondria so that they can undergo the electron transport chain. And that's what I'm about to do. Now, this NADH here, they're going to come. So inside the cytosol, remember this is the matrix, eh? So inside the cytoplasm, you have oxaloacetate. Now, oxaloacetate is going to react with the NADH. Right? Now, NADH is going to unload the electrons to oxaloacetate. So that is converted to NAD+. Now, oxaloacetate is going to be converted to malate. Now, malate now has the capability of penetrating to the mitochondrial membrane. So, malate is going to come and get inside the mitochondrial matrix. Now, remember it has the electrons. So, it's going to unload the electrons. So, let me clear that from this space. Let's cross the median. Now, it's going to get inside the mitochondrial matrix. So, it's going to unload the electrons. So, we're going to have NAD+, is going to pick the electrons, converted to NADH. And now we can have the NADH so that they can go to the electron transport chain. So, malate after unloading the electrons is going to be converted to oxaloacetate. Back to oxaloacetate. And now, we need to get rid of this oxaloacetate inside the mitochondrial matrix. What are we going to do? So, we have our oxaloacetate here. Now, oxaloacetate is going to react with a molecule. What's the name of that molecule? Glutamate. When oxaloacetate reacts with glutamate, now they are going to react. Glutamate is going to be converted to alpha ketoglutarate. Alright, this is glutarate. Alright, then oxaloacetate is going to be converted to aspartate. Alright. Now, aspartate can now get back into the cytosol. So you have the aspartate outside the matrix of the mitochondria. So inside the cytosol, now we need to get back to oxaloacetate so that it can continue taking up the electrons from NADH, getting into the mitochondrial matrix. So you have aspartate is going to react again with alpha ketoglutarate. Now, when they react, we are going to form glutamate. Alpha ketoglutarate is going to be converted to glutamate. And now aspartate is going to be converted to oxaloacetate. Now, we have it here. Alright, so this is the way or the mechanism by which the body gets the NADH generated from glycolysis into the mitochondrion so that we can undergo the electron transport chain so that we can generate energy. But remember from TCA, it already occurs inside the mitochondria. Therefore, there's no need. So already they can now go to the electron transport chain so that can generate energy in form of ATP. And mostly from one molecule of glucose, you always generate, you can generate 38 molecules of ATP. Some you can find 32. Now, you may wonder why 38, why 32? So this just depends on the values that you use. Maybe when you use one molecule, one NADH gives us how many molecules of ATP and one FADH2. So some books use 1.5 to 2.5. So it will just depend on the value you used. But don't worry, whenever you understand the concept, it's just you're good to go. Now, the end product is ATP. We've learned about the uncouplers and the inhibitors. So thank you so much guys, and whenever, suppose you find this video helpful, please subscribe to my YouTube channel, like and share this video. Thank you so much.